começo era pra ser só beijo, mas tu me abalou e virou meu desejo.
Welcome to my video. In this video, I'll take you along with me as I share how my professional practice went in the beautiful city of Rio de Janeiro, Brazil, during April and May of 2025. Over the course of 2 months, I had the incredible opportunity to work with several professors and discover a new culture, the vibrant and warm Brazilian culture. I learned so much thanks to a well-organized professional practice hosted by the University of Unirio. This internship was divided in 2 main parts. In the first part, I explored 5 different research labs, each time with the different professors and each for 2 days. Let's start with the first one. Here, I joined Professor Cristina at the Lagoa Rodrigo de Freitas, a stunning lagoon in the heart of Rio, just next to Ipanema. We studied their zooplankton in their natural environment. As you can see here, we collected samples of zooplankton directly from the lagoon, which we later analyzed under the microscope back in the lab. A few days later, I joined Professor Rodrigo on a field trip to Arraial do Cabo together with Lucas, a second-year IMBRSea master student, and Luciana, a Brazilian student working under Rodrigo's supervision. The purpose of this trip was to help collect behavioral data on green sea turtles. We spent about 2 hours snorkeling and filming the turtles in their natural habitats to support Lucas and Luciana's studies. I now let you enjoy some footage we took. The next lab I visited was with Professor Tatiana Maria, who works with meiofauna. We went to Praia Vermelha, a beach just beside the university, where we collected sand samples. In the lab, I first filtered the sand samples and then prepared microscope slides. After that, I observed them under the microscope, first by sketching what I saw, and then identifying the different meiofauna species present in the samples. And finally, the 5th lab I visited was with Professor Rodrigo from another university in Rio. He specialized in cetaceans. He invited me on a boat trip with his team in Sepetiba Bay for a field trip entirely dedicated to photographing dolphin dorsal fins for photo identification. These images help track individual dolphins and understand their movement patterns, like where they feed and where they rest, both during day and at night. It was a beautiful sunny day and we ended the trip with a refreshing swim near the rocks in the middle of the bay. A perfect ending to a long day of field work. Now, let me tell you about the second part of my professional practice. I chose to work with Professor Wanderson on a new research project focused on green sea turtles. The main goal of the study is to determine whether these turtles can serve as biomarkers of environmental quality based on their site fidelity. As you can see here, we are capturing a turtle on the first research site, a polluted area. To support this research, I was in charge of developing a protocol to analyze the biofilm as well as the phytoplankton. I gently scraped their carapace to collect the biofilm, then placed the samples into a tube for later analysis in the lab. My goal was to test a methodology, first for collecting, then for analyzing samples of biofilm, phytoplankton, and macroalgae. Here's the beautiful setup of the lab where I worked with Professor Wanderson. After collecting the samples, I labeled each tube to keep everything organized and traceable. Then, I prepared subsamples to get the right volume needed for the flow cytometry analysis. This is a quantitative and blind analysis with results that need to be interpreted afterwards. Here we are at the second research site, Praia de Itaipu. As you can see, the methods used to capture the turtles here is quite different from the first site and required more people. Once again, we applied the same methodology and we collected biofilm by gently scraping the carapace, just like at the first site. Before that, we had also taken water samples to analyze the local phytoplankton and microalgae. After a full morning in the field, we share a lunch break as always with the team. This was a typical dish from Rio. Back in the lab, I followed the same process, labeling tubes, preparing some samples and getting everything ready for flow cytometry. Together with Wanderson, we reviewed the results and tried to interpret this analysis as accurately as possible. After 4 weeks of hard work on this new research project, I presented my results in front of a scientific audience. We wrapped up this incredible professional practice by sharing a few beers with the professors and students involved in the project, all while enjoying an amazing sunset over the beautiful city of Rio de Janeiro. I'd like to thank Professor Wanderson for everything he taught me and also express my gratitude to all the other professors and students who contributed to the success of this incredible project.